Most people think, well, gee, water is such a simple molecule and it's all over, and scientists have been studying it for years and years. Everything about water that could be known must already be known. But in fact, water does so much. It participates in virtually everything that goes on in every one of your cells. If you line up all the molecules in the cell and just start counting, 1, 2, 3, more than 99 out of 100 would be water molecules. The reason being that they're so small, and to make up that 2/3 volume, you need a lot of water molecules. So, to state, as in biology is often stated, that the water molecules merely are background carriers. They don't do anything. They just sit there. It's a kind of, intellectual arrogance. There's no junk in the body. Everything is there for a purpose. Penetrating toward the truth is exciting for almost everybody. I think we all have that desire, and sometimes we're exposed to science, and the science seems so elusive because it seems so complicated. But I guess I've developed some philosophy over time. I've been doing science for more than 50 years, and I guess a half century is enough to have developed a philosophy. And my philosophy is science is really simple and anyone can understand it, even me. And if it looks complicated, or if it seems complicated, it might be, and there's a good chance that it's not correct. I think excuse my nose for a moment.
Of course.
I think there are a lot of concepts that have made their way to the textbooks that may actually be not true, and
that's a pretty bold. That's a bold place to start from.
Well, why not? Why not be bold? I guess what I prefaced my remarks with the idea that I've been doing this for a long time.
Yeah.
And,
well, it seems like science necessarily has to break down what came before in order to move forward.
Well, actually, yeah, that's a really good point, right? Because if you are developing an understanding of something, let's make sure we have the same definition of science. For us, it's mechanism. Explanation how a phenomenon happens.
Exactly. I'm with you on that. You can observe something in nature, and the question is, what's going on here, and we need to find out. And unfortunately, there are foundations that have been developed, and if you try to build an edifice on a weak foundation, it doesn't hold up. And this has been the issue. There are many weak foundations out there. And if you try to build on it, everything becomes complicated. You need struts and Band-Aids to cover the gaping wounds, and ropes to hold it in place. And it's not beautiful.
Why is it so hard to work on the foundations themselves?
You guys are just full of insights and great questions. So I love it. Yeah, the reason it's hard is the system of doing science. So what do I mean by that? I mean, for most science is done by academics, and academics work in institutions. And like myself, I'm at the University of Washington, and in order to run a laboratory to do research, you need money. So I'll give you an example. As an academic, suppose I have an idea. Okay, and my favorite example is my idea is that the Earth is round, And everybody knows that it's flat because all you need to do is look out there and, you can see it looks flat. And if you happen to be fortunate enough to be near a body of water, you look out, it looks flat, no bumps, nothing, just absolutely flat. But you have the idea that, maybe the Earth is round. And the reason you have the idea, despite what everybody thinks, is you've seen satellite photos, right? And hey, it looks curved, and in some cases it looks like a sphere, and you think, by golly, I think the Earth is round, despite the fact that everybody thinks that the Earth is flat. And you've even gone one step further. You've taken a flight. You've left from wherever you reside. In my case, Seattle, let's say. You come to Seattle, you take a flight west, and you land in Honolulu. And then you go on for farther west, and you land in Tokyo, and you go farther west. And you keep going, and you wind up where you started, back in Seattle again. Keep going. And you're very conscientious. So you look out the window and you think, with some degree of cleverness that, if the Earth is flat and you're able to return to where you started, the Earth must be a cube. There must be some edges, right? Well, yeah, and you look out the window and you're extremely conscientious. You even don't stop for meals because you really want to look out and see. And you never see the edge of the Earth. You never see those. So you've got two pieces of information now. You've got your personal evidence that you look for the edges and you can't find them. And the satellite photos seem to show it's a sphere. So you're thinking, this is really important stuff, and you want to study the shape of the Earth. So what do? You go to one of the funding institutions and you say, hey, I have some, what's called preliminary evidence. Those two pieces of evidence that we're discussing, and please, could you give me some more money because I want to set up a laboratory to investigate the shape of the Earth. If it's really flat, as you guys think, well, that's okay. But I think it might not be flat. It might be round. And if it's round, it impacts so much of our thinking that I want to study it. So please give me some money because I can't do anything without a little bit of money. So it goes to the gatekeeper. And the gatekeeper sees an application from the two of you, perhaps, and this looks pretty radical, pretty important. And they want to make sure, the gatekeeper wants to make sure that it gets proper review because gatekeeper doesn't know you guys. And, are you charlatans or are you for real? So they recruit the very best people in the field of the shape of the Earth to review your application for money. So who are these people? Well, these people are the experts in flat Earth theory. And the question is, well, how is your application for money going to be received by these reviewers?
In
Not
theory,
very well.
Well, in theory, this is something that we talk about a lot, which is the responsibility of the individuals within a system to make it functional. Because if the system was filled with people who looked at their theories as being one possibility, something that needed to be re-examined periodically, then they'd look at that grant application and they'd be like, well, yeah, let's look. Maybe we're wrong.
Well, I'm with you on that, but try to find such people. You guys would fit into that category perfectly, but most people, it's human nature, and we want to protect ourselves. And so, if somebody came to challenge my ideas, my theories, I'd hope that I would be open to it. But But we're human beings, and we want to protect our turf, so to speak. And if somebody challenges my ideas, one side of me says, well, of course, we're looking for truth, and challenge is completely appropriate and necessary. But the other part of me says, oh boy, my career, I made my career on XYZ, and somebody is challenging the very foundation of XYZ, and therefore, if they're right, I'm wrong. And if I'm wrong, I'm going to have real difficulty in the future getting money for my scientific endeavors.
Oh, and you'll also look bad because you'll have been pushing a bad theory all along, right?
Yes and no, because I've got a lot of colleagues. If I'm a flat Earth person, most of my friends are also flat Earth people, and they're thrilled if my vote is to reject this application. They're very happy about it. On the other hand, if I'm sitting around the table and we're doing the review, and I say, well, wait a second, all of us are flat Earth people, but maybe there's some truth to this, and if so, I'm really vulnerable because all the colleagues sitting around the table are going to say, well, then how do you explain X and how do you explain Y? And it puts me on the spot, and it can be really embarrassing if I can't answer their questions objectively. So the easiest for me is to say, oh, well, this application is pretty interesting, but gee, I wish they would only have provided or told us the statistical analysis that they're going to use to explain their data. Tell them to come back next year with another application, etc. So
has this happened to you? Sorry, has this happened to you?
Yeah, it's happened, but
probably happens to everybody to some extent.
Yeah, it does, because there are so many applications and not enough money, so you have to choose the ones that are the best.
Well, I mean, not about the grants applications specifically, but about the sort of the dichotomy of going against consensus. Like,
what about the dichotomy? I'm sorry, what's your question? I missed it. Have
you been in that position where you're the person coming around and suggesting that maybe the Earth is round and everyone else?
Essentially, that's all we do in our laboratory is exactly that, because we're interested in finding truth. And as you pointed out earlier, it's difficult to find truth without often challenging the existing concept or ideas of where truth really lies.
And your
Otherwise, there's nothing to do.
Your Earth is round idea is, just to put it in a sentence for our audience, if they're not already familiar with your work, you've got a lot of great talks out there and people can check you out on YouTube and stuff. But the basic idea, if I can try to paraphrase it, is that water, it sort of forms these composite structures which can be treated as objects inside of biological systems.
Yeah, I think that's an interesting way of putting it.
How would you put it?
I have new respect for canine creatures.
That's the second time we've gotten dogs in like 2 days.
Closest Earth animal.
Yeah, exactly.
Yeah. So water is what we study, and most people think, well, gee, water is such a simple molecule, and it's all over, and scientists have been studying it for years and years. Everything about water that could be known must already be known, and
it's sort of a bystander.
Yeah, bystander, or as the biologists like to say, it's like the bathing medium, the medium that bathes the more important molecules of life. It's boring. It doesn't do anything. It just sits there.
It's the ether. It's the ether of biology. It's just kind of there.
You might call it, but some people think the ether is actually very active.
That's true. It's the passive ether of biology.
Yeah, right. I think that's an interesting way of putting it. But in fact, water does so much. It participates in virtually everything that goes on in every one of your cells. So everybody knows that our cells are filled with water. We're 2/3 water, but not many people realize that that if you translate that volume fraction 2/3 into the number of water molecules, that is, if you line up all the molecules in the cell and just start counting, 1, 2, 3, more than 99 out of 100 would be water molecules. The reason being that they're so small, and to make up that 2/3 volume, you need a lot of water molecules. So, to state, as in biology is often stated, that the water molecules merely are background carriers of the more important molecules of life, it's really hard to digest that, because it says that 99 out of 100 of the molecules inside your body, be it human or canine, don't count. They don't do anything. They just sit there. It's a kind of intellectual arrogance to say that.
Well, that's what humans said for a long time about, what is it, junk DNA?
Oh yeah, junk DNA, same sort of thing. There's no junk in the body. Everything is there for a purpose. Everything does something. So
you think these waters are sort of doing more than just carrying along other molecules? The sort of the water molecules themselves are acting as signals, as a structuring entity, like a structured entity.
Well, yeah, a structured entity. That's exactly it. Structured entity. And so maybe I should just say a few words about the structure, because we think about water as having three phases. And one of them is structured. That's ice. That's the solid phase. And then we have the liquid phase, and then we have the vapor phase. But what we discovered is that there's another phase, and this phase we refer to it by several different names, and I apologize for that. Let me just say what they are. Yeah, one is the fourth phase. Okay, another one is exclusion zone water, and we call it exclusion zone water because early on when we discovered it, it turns out that this kind of water does have a definite structure, just like ice, but not ice, not exactly. And as it forms, it excludes all kinds of stuff from it. It forms a pure crystal, and in order to form a pure crystal, it's got to get rid of any impurities, just like ice does as it forms.
It's very viscous.
It's very viscous, right? And it basically. So we call it exclusion zone, or for short, EZ. And that works really great because it's easy to remember. And, the problem, though, is it doesn't work in most countries because they don't use the term Z, they use the term Z. So you've got EZ water instead of EZ water, and EZ is more difficult to remember. EZ is easy to remember. So at any rate, whether we call it EZ or EZ or fourth phase water, it's this water that's different from other water. And it's not merely a laboratory curiosity. It fills every cell in our body. And it's necessary for, it's difficult to say every reaction that occurs because we haven't examined every reaction that occurs. But
this water... Oh, sorry, go ahead.
No, but some of, or many of the most important things that go on in cells, water is intimately involved.
Can you say more
Without
about
that?
that?
Water
the
doesn't
structure.
Work. Sorry?
So in terms of chemistry, I think that's pretty well established, it seems like, for earth scientists. But this idea that there is another phase of water that's participating in processes, can you sort of... Well, just to reiterate, this water is sort of occurring at the interface with the cellular material, if I understand correctly. And then my question is, like, how does that actually affect different processes within the cells? Like,
is sodium-potassium transport affected the same way that, say, protein transport is?
Well, okay, so your questions are a little bit different from one another, but let me just say, in the essence of of how that water works is, first, your cell is filled with it. Secondly, when the cell works, cells sometimes are sitting there sort of doing nothing, like lying on the beach in Hawaii or something like that.
Really?
And it's, well, no, not exactly, but they absorb energy that way. I'll talk about that in a moment if you ask me. There is a kind of energy that's required, and it comes from the environment. But then when a cell is in its so-called resting state, the water inside the cell is EZ water, or fourth phase water.
All of it, or just at the interfaces?
No, it's all over the cell. But I mean, the cell is full of interfaces because you've got a lot of solids inside the cell, and next to each solid is EZ water, and the solids are so closely packed that essentially all the water is EZ water. So then a signal comes along. Let's say you could be a nerve cell, a secretory. Let's say you're a muscle cell. You're not contracting. You're sitting there, and the water is EZ water. And then a signal comes along and says, "Oh, time to contract," because you're a frog and there's a fly and you want to catch the fly. And in order to catch the fly, you got to move. And in order to move, your muscle needs to be activated. So it gets activated. And the first thing that happens is that the water undergoes a transition from EZ water. Also, it's called structured water by many. EZ water into ordinary water. And the ordinary water then is the proteins that actually do the work are sitting next to the water because each protein is surrounded by EZ water. And what happens is as the water undergoes the transition, so does the protein. And the protein goes from one state, so-called extended state, into another state, so-called contracted state.
How's that signal transduced?
Well, it's not exactly transduced. It's a so-called phase transition, and the phase transition involves the fluid, the liquid, the water, and involves the solid, the protein, and both of them together undergo this transition.
Is it sort of like ice skating? Like there's pressure exerted on the ice, or I guess this isn't technically ice, but it's a sort of solid-ish form of water?
Yeah, I'm trying to understand
what causes that phase transition.
Yeah, exactly. So you have, because we have a story of the frog, right? The frog sees the fly and wants to catch the fly.
In the case of the muscle, it's a trigger of some sort. In the case of the muscle, it would be that a nerve is sending a signal
like electrical?
to the muscle.
Mm-hmm.
It's time to contract. And the first thing that happens is the water begins undergoing a change.
Like before the calcium is dumped?
Well, the calcium is less clear. The calcium may be dumped.
I guess I'm just trying to understand what causes that change in the water in the first place.
So you have structured water around the protein. Protein is in state A, and then there is a transition to state B. And the question is, why does the transition occur?
What's the
driving
it?
Occurs in the case of let's say a nerve ending, which may release, say, acetylcholine or something to signal to the muscle, that initiates it. And as soon as the water begins undergoing this transition, it propagates. It's a natural process to propagate, and it propagates quickly, and it propagates throughout the cell. So the entire cell undergoes this transition of both the water and the protein.
But it's like a chemical process initially?
Yeah, like the acetylcholine.
Is it like salting ice in the winter on the roads or something?
It's a physical chemical process that takes place. The chemistry of the water actually is changed. Due
to the neurotransmitters?
Sorry?
Due to the neurotransmitters? It's like there's
It starts at the neurotransmitter. It starts with that, but it could be initiated many different ways. And one way is to to apply calcium if you have an isolated cell, not in the body, but in an experimental chamber that you're looking at. You can apply a little bit of calcium, and that calcium will be the same trigger that for example, acetylcholine would be.
Gotcha.
You can get many agents to trigger it. Even the change of pH could be a trigger.
I get it.
Yeah. And so once the trigger occurs, all this occurs regeneratively. It permeates the entire cell. And so the cell then contracts, because it wouldn't do any good if part of a cell contracts, but the rest of the cell doesn't contract, because then, you'd have one part of the cell pulling on another, and that's not what you want. You want the entire cell to undergo this transition to contract, and then it's effective.
Makes sense.
And
So
what's the nature of the structural change in the water?
Well, the nature is it goes from the EZ water, or a fourth phase, which I haven't told you about, but let me tell you, to regular water, and then at the end of the contraction, back to the initial state. So this kind of EZ water has a structure that is entirely different from ordinary liquid water, because the water actually undergoes a chemical change, but not so different from ice. So ice has a hexagonal kind of motif, and so does EZ water. EZ water, if you have just think of a flat surface, and water is next to that flat surface. What happens is that the molecules of water that are in contact with that surface undergo the transition, and the transition forms a sheet, a hexagonal sheet. And that sheet, actually, the chemical structure is H3O2. It's no longer H2O. And it has negative charge, typically, by the way. It's not neutral. So the first sheet forms. And if you were to look at the sheet, you'd see hexagons consisting built of hydrogens and oxygens. And then what happens is that the next sheet forms. And the template for formation is the already existing sheet. And then the next one, and then the next one, and so on. And it can build according to our experiments.
So it makes sense that if you disrupted the first layer chemically, that the next one would follow, and so on and so forth. And you'd sort of lose this integrity throughout the cell.
I think that's correct. Although, experimentally, approaching a single layer or even a few layers is not so easy, because these layers are, if you think of a membrane, which is, say, 10 nanometers in thickness, this is, 30 times thinner.
Yeah, you're operating really on the edge of what can even be observed.
Water is what? Is the water just like a third of a nanometer or something?
Well, a typical molecule is a third of a nanometer, roughly.
So these are really little structures.
And
they're really tiny.
Are you using AFM to look at these, atomic force microscopy, or how are you?
Well, we have long ago, but we don't use it on a regular basis. We have used micro electrodes, and these electrodes measure the voltage, electrical potential, and you need two of them because you're measuring a potential difference. So what we've done in the past is we stuck one in the exclusion zone and another one in the region beyond the exclusion zone, and you get a potential difference on the order of 100 millivolts or something like that, where the EZ is negative.
So there's some capacitance effect of this viscosity shift or something?
I like to think of it rather as a battery rather than a capacitor, but the two are really so closely similar that one can substitute for the other. So you get a charge separation. This is the essence, really, of what we're talking about. The EZ is typically negatively charged, and the region beyond the water, inside the water, is positively charged. And we measure that several different ways.
So it stores a lot of energy, basically. The idea is that it can do a lot of work when it's signaled to do so.
Totally, yeah. This is a really important aspect. And inside, your body, although I'm not sure how your bodies operate, maybe a little different from the rest of us, but
we'll tell you when we get there.
Okay, well, thanks. Yeah, so if we have this inside our bodies, it means we have batteries inside our body, and you could think of the batteries maybe doing nothing, or you could think of them as actually storing and delivering energy. And of course, I think the latter. So I think inside our bodies, it's not just the usual ways of procuring energy through ATP and such. It's that the water itself stores electrical energy, and this electrical energy can be used to drive many processes.
Well, this is actually a fascinating detail to be added to the canon, right? Because you have, let's look at proteins. There's this shape change that happens when a protein binds a molecule.
Like a spring.
Like a spring, yeah.
Totally.
And I've always wondered about what drives that, because you look at these proteins and you have this huge glob of stuff, and it binds this teeny tiny little thing. And they say that it causes a big swing in the shape. And so if there's a layer of this easy water that's holding it in one conformation, and then the molecule that comes through that is bound by the protein destabilizes the easy water, then you'd have a relaxation event like the one you're talking about, and you'd get a conformational change in the protein.
Absolutely, but then you, at the end of the story, you have to drive it back to its initial condition.
Well, you remove the chemical, right? The protein lets go of whatever it's holding on to, and then the structure reforms. I can imagine, this basically, the water's always there, right? And if the chemical that comes in destabilizes the layer of the water, and then it leaves because the protein spits it out, well, then the water would reform, right? But you're saying that it's a different chemical formula of water.
Well, I'm not so sure it's exactly that way, because in order to create order, you need energy. You see, and as we were talking about this process, it starts ordered. The water is ordered, and it has this regular structure. And then
but it's ordered by the protein structure, right?
Yeah,
it's ordered by the sort of hydrophobic, hydrophilic shape of the protein.
The energy came from elsewhere in the cell to form the proteins in the first place. Like the proteins, energy was taken, but it was in the formation of the proteins, right?
Well, I'm not. You might be right. I might be wrong, but I look at it at a different way.
How do you see it?
Well, the potential energy is there. It comes from order. From the water is ordered, the protein is ordered, and then this order all collapses as the water undergoes the transition from order to disorder, and the protein also undergoes a shape change. But you need to drive it back to where it came, to the initial state, because this happens not once, but it keeps happening. And that's where the energy comes in. That's where you need energy to separate charge, to create order. And then you're back again. And so
But this is like what ATP is used for.
Well,
right?
It could be ATP, but not necessarily. So, and I go to Gilbert Ling, someone whose name I haven't mentioned, but he was a pioneer in the idea. The late Gilbert Ling, he's passed age just less than before 100 years old, and he did amazing work demonstrating the ordering of water inside the cell. And he looked at ATP and a little bit, but he refers to a critical paper that I need to mention. So many years ago, so a group of scientists said ATP has a special high energy bond. And that high energy bond is used to power lots of things. A year later, some physical chemists said, no way, you guys made an arithmetic error, simple mistake. There's no high energy bond. All the bonds are the same. And Ling talks about this in his books and his website. I hope his website still exists. I'm not sure. gilbertling.com. He talks about it. And nobody's ever addressed that issue.
Well, so, I've read lots of papers that have not necessarily addressed that issue openly, but they have started to talk about the fact that there's nothing special about that bond.
Well, that might be the case. So this is an open question right now.
Yeah.
Somebody needs to and if it's not ATP, as Gilbert Ling argues strenuously, it's got to be something else. And I'm not suggesting that something else is necessarily completely the energy that structures or orders the water, but I think it could be. There are lots of people who get on without eating. The so-called breatharians, and there's good evidence. This is real. It's not a fiction. And so, where do they get their energy? Well, I think it's possible that they get their energy the same way that the water gets its energy, and I haven't mentioned it, but that is through light. Yeah, and not visible light so much, although some of it may come from there, but infrared light. And in the case of infrared light, the wavelength, as you guys are sophisticated, beyond a longer wavelength than a visible light. And the experiments we've done show that the energy, the best wavelength, or the most effective one, is about 3 micrometers into the infrared. So where does this energy come from? Well, the sun, to start with, something like half the energy of the sun that reaches us is in the infrared region. So effectively, it's sunlight that does it. But, sometimes, especially where I live in Seattle, the sun doesn't shine, but we still survive. Our muscles can still contract. Our nerves can still conduct. Our secretory cells can still secrete. And what happens is that all the structures around us absorb energy from the sun and then re-radiate. So everything is radiating infrared energy, and you can demonstrate that easily with an infrared camera. Completely dark, you can't see anything. Your cell phone camera doesn't show anything. But you whip out your infrared camera and you get a beautiful image in the dark of everything around you, because everything, instead of radiating ordinary or reflecting ordinary light, radiates infrared light, and your camera lens and sensor can pick it up. So it's always there, which means that since that's the energy that's required for the buildup of easy water from ordinary water, we got that energy. So to summarize, infrared energy is absorbed by the water, converts the water into EZ water, and that water has a lot of potential energy. Whether it's all the potential energy in our body, I wouldn't be so bold as to suggest that, but we probably get substantial amounts of energy, electrical energy from this water, from this easy water, or you could call it electromechanical energy if you wish. But the source of the energy initially is infrared light.
So it's like the water molecules, they get all this buzz from the light that's flying around, and then the walls and all the surfaces within the cell sort of tell those charged up molecules where to go. Those waters, they're like, go stand over here, and then they just sort of build on top of one another.
But there's a chemical transition here that is caused by the infrared light.
To the hydronium.
The energy, or not exactly. The energy is supplied by the infrared light, but the surface is what counts. The surface of the protein, for example, the surface is charged, and the water molecules are lying right next to it, and with the right charge distribution on the surface, which is characteristic of biological molecules, the water can undergo this transition. It can't always undergo it. If it's sitting next to the wrong surface, it won't undergo this transition. And the wrong surface would be, for example, hydrophobic surfaces like Teflon, for example, won't do it. But hydrophilic surfaces, which characterize pretty much most surfaces, they tend to be hydrophilic. And most of those surfaces will permit or facilitate the transition. So the energy comes from the light, but the catalyst, you might say, is the charges on the surface of the solid.
That's fascinating. So the surfaces are sort of the template, They sort of set up the chemistry, and then the water's got all this wiggle in it already, and it sort of just does what it's told and uses all that light energy to power its motion.
Absolutely. That's exactly how it works. You guys are really astute.
So that's really
work in my laboratory.
We got a lot of work to do on our own. But my real question, to take this back to where we started, is why on earth would that not be an attractive proposal? And if it isn't, why is taking on such high-risk projects, is there a way to incentivize these sort of higher risk ideas when they maybe go outside of what's traditionally understood at the fundamental level? How do you incentivize grantors to take on risks like that?
And maybe it has to be outside of the institutions? Is that possible?
Well, yeah. So, okay, we've taken a step in that direction outside of the institutions, as you say. It's called the Institute for Venture Science. And recognizing the difficulty, we put together an organization whose mission is to do exactly what you guys are suggesting. We're trying to get money. We got some, but not enough to fund scientists who are proposing to examine fresh paradigms where the existing paradigms don't make sense anymore. As we all know, it's hard to do that. It's hard to get the money, but this institute will accept only proposals that do this. And the vetting is very serious. We've gone through 200 pre-proposals, invited 15 proposals from that, and selected 5 of them for funding.
What are the topics? What are the topics that you're looking at?
Well, so I'll give you an example of one of them that is not yet funded, but is one of them on the list, and it's a cancer curing proposal. So, as we all know, President Nixon, almost a half century ago, declared war on cancer, but the war has not yet been won. There are some advances, but
didn't he also declare war on drugs, or is that somebody else?
Was it Vietnam?
Yeah, it was a lot of wars that he declared.
Yeah, he's not good in the War Department.
And that's right. Yeah, so we can have our opinions, but political opinions and otherwise, but cancer problem remains. And my old wife, unfortunately, a year and a half ago, passed from ovarian cancer, and
I'm so sorry.
And many of the anti-cancer therapies have not changed a whole lot in the past 40 or 50 years. There are some advances, but we haven't solved the problem. Nobody has said, okay, well, this guy has a proposal to solve the problem, and it's a really interesting proposal. It's completely unconventional, yet he's demonstrated with mice amazing results. So I'll just tell you, because it's kind of interesting. His proposal, his experiments. He collaborates with people who are using a mouse model. This is a very standard model. You inject tumor material. It's a mammary tumor. You inject it in mice, and if you inject it, the tumor grows, and within, I think, 35 days or so, the mouse dies. And the mouse dies because the tumor grows so big that it presses on all the other organs. It's 100% lethal.
Yikes.
So what this guy does is he collaborates with people using this model, and they're trying drugs and other expedients and seeing how they work. So he takes a cage full of these mice that have these tumors sticking out. He puts his hands around the cage, and he goes through a mental exercise. And his mental exercise is thinking through a list of positive thoughts. They're actually images of positive thoughts. So positive thought might be embracing his pets, like you guys, right in front, and feeling good about it. It might be shaking hands with the King of Sweden when he gets his Nobel Prize. I'm making these up, et cetera, et cetera. And he goes through a list of 20 of these, and in each case, it's an image that he has. So he's thinking about the image and feeling good about it, feeling positive. And he runs through, and he starts again at the top, and he runs through, and he runs through, and he said it's boring. And his cure rate is practically 100%. And they're really cured.
From xenograft tumors? That's amazing.
That's incredible. Seems like he'd be in high demand at clinics worldwide.
Has he published this?
You might think so, but of course...
Can he teach it to other people?
Yes.
Whoa.
He can teach it, even to you guys.
Has he published
canines
this?
work just fine.
I like this idea that we're a race of super intelligent canines coming back to Earth. I appreciate that.
Has he published this?
Yeah, has he published this?
Yeah, he's published it, and you could check him if you like. And his name, I guess I don't mind.
I got a mole I have him look at, actually.
His name is Bankston, Bengston, William Bankston.
All right. We'll put it in the description.
And he's a really interesting guy. The problem, of course, is that the whole pharmaceutical industry, they're not enthusiastic at all.
I wouldn't say...
And they have a lot of clout.
Yeah, that's not really
a
conventional... That's not a conventional treatment approach.
Hard to put that in the bottle.
In fact, it's so unconventional that a lot of people would just immediately turn off. But if you read his papers, he does it not only on mice, he does it on humans too. But humans, if he has a success with humans, it's dismissed as a placebo effect. And, how are you going to argue against that? It's not so easy.
Well, the placebo is pretty incredible by itself.
I didn't hear you.
The placebo is that's quite an incredible thing all by itself.
It is.
That's a whole other discussion.
Indeed. Yeah, but this is, and I just want to mention in addendum that the mouse, in this case, is actually cured so that if you inject the tumor material again, no cancer develops. It's protected.
That's incredible.
So what's the mechanism here?
Okay.
And of course, he would like to find out the mechanism of what's going on.
Sure.
So this is just one example.
But you said you're funding 5 proposals. Can you share all of them?
We're hoping to begin by funding 5 proposals, or 1 or 2 of them. And
sure.
But here's the trick. You see, we know that any kind of proposal that's out of the mainstream, or any scientific endeavor, is going to be attacked by the mainstream. Oh, Bankston, he's a crackpot. Don't pay any attention to him.
Well, that's basically crackpot is like it's a lethal word in science. And it's thrown around just when people kind of don't like the work, I think.
Unfortunately, right. And how do you defend against it? You say, oh, no, you wave a flag. I'm not a crackpot. It doesn't work. So,
exactly.
Once the mainstream... That
sounds like something a crackpot would say.
Yeah, sometimes there's no evidence behind it, but merely someone of some significance standing up and saying, oh, I don't believe that stuff is nonsense, and then you're finished. But at this institute, we defend against that. And the way we do it is this. If we fund Bankston, we will fund, assuming the money is available, we will fund up to 12 groups, independent groups who are using pretty much the same approach as Bankston, maybe some variant. And if suddenly there are 12 groups who are reporting success using this method, it becomes difficult to say crackpot anymore because what does it take to convey truth repeating in other laboratories is the general standard. And
yeah, I mean, I think that it requires a pretty intense process of controlled study, right? Double-blind, controlled, placebo-controlled studies.
Yes.
Is the gold standard, and they have to be carried out in a variety of different laboratories, different universities, even. It's hard for me to imagine that the Crackpot label could be washed away if it wasn't somehow being considered within the institutions themselves, because that's an institutional label. Crackpot.
Well, why do you say institutional label? I mean, I'm a professor at the University of Washington, an institution. Institutions don't take stands.
No, it's the people within them, right? So if someone is... It's hard to imagine a scientist that isn't associated with an institution like the University of Washington labeling another scientist as a crackpot.
I think what Quinn's saying is it's like a power word. Like, the only people calling anybody else crackpots are people who are defending some pretty well-established position in the first place.
Yeah.
Well, sure. Yeah, that's who it comes from. These are the people who are motivated to use the label crackpot. And they're basically, trying to defend their own positions by doing that. And they have, the weight of numbers on their side.
Yeah.
And so by funding multiple groups, we defend against that. And thereby, therefore, I really hope we can be successful. We're looking for people. This is privately funded because governments don't fund operations like this, and we're looking for people who have done well in their lives and who are looking for a really meaningful way to donate some of their money to doing something meaningful for society. And I can't think of a better way to do this, because there's so much that remains to be discovered that nobody ever even considered or thought of. And sometimes, or almost always, new fundamental scientific observations lead to new technologies that nobody could ever have dreamed of, right? The best example is the transistor. It was like 60, 70 years ago, some impurities and semiconductors led to what we're doing right now. Who could ever have conceived of something like that many years ago?
And it seems like private research institutions kind of were the norm at some point. Like, this is kind of a new idea that the public funds all the research, at least in the last 100 years or something, from what I understand.
Yeah, but just think the last 100 years or so, well, at least the last 50, 60 years ago, but 100 years ago, there was very little money coming from governments. Funding, if wherever necessary, came either from a benefactor who believed in the scientist, do what you want, I'll give you the money, or a wealthy family. See, and
I like this idea of bringing back the patronage system as a mechanism for dealing with wealth inequality.
Basically, I like it too.
Basically, have rich people have an app where you can send them an application for a project.
Well, that's the story of the Enlightenment, the first one anyways.
Is it?
From what I understand, there was a lot of patrons and in the rational, whatever it was.
Well, the Renaissance was basically
that's what I'm thinking of.
The Medici family funding artists, musicians, and what a wonderful period that was of contributions. And we're now not in a period like that, but wouldn't it be wonderful if we were? The science, the arts, together would be fantastic. So we're hopeful that we're going to be attracting people with some means who would like to support this kind of endeavor. It's not cheap, unfortunately. We're not talking about a few $100 here or there. A typical scientific endeavor or project per year could easily involve a couple of $100,000 easily.
Sure.
And typically these scientists need multiple years. It can't be done in 1 year. It takes maybe 3 years or 5 years. And so we need appreciable funding, and we're looking. By the way, if anybody has interest, just contact me, and or our website is ivscience.org. IV is an intravenous, or Institute for Venture. ivscience.org. It's all there, and I'm freely available to discuss stuff like that. It's really necessary. Something needs to be done to change. And the evidence for that, I'm sorry, I'm ragging on and on.
No, go
is
on.
If you ask yourself, how many scientific revolutions have you witnessed in the past 20 or 30 years?
Or cultural. Everything's stuck.
Or cultural. Yeah. How many? Right. And I'm not talking about technological revolution. I'm talking about fundamental scientific, and you might add cultural to that. And it's really hard to identify them. But 100 years ago, when there was no institutional funding, these were occurring at a furious rate. We have, especially in physics, practically every year something was coming out. So we need to restore that once more.
That's a beautiful vision.
I love that.
Well, thank you. Yeah.
So I know you need to run. We would love to talk to you again in the future sometime and get deeper into this and see how your initiatives are going.
I want to hear all the IV proposals.
Yeah, I'd like to hear how all that turns out.
Oh, okay. Well, we're happy to do so. I especially enjoy interacting with the canine folks.
We didn't even get to the Brazilians. That'll be next time.
We can talk again another time. Sure. Absolutely. My pleasure to be with you guys. I love your unique format.
Thank you, Dr. Pollack.
Thank you so much.
Sure. My pleasure.
Take care.
Okay. Take care.
Bye.
Bye. Bye.